Oh, good work, sweet pea. Hello, everybody. Thanks for coming back into the icebox. We really appreciate you all sticking around for our last talk. This is going to be a great one. Rob Robbins from the Florida Museum of Natural History is going to talk to us all about fish. But first, before we do that, we wanted to take a minute to bring up our team. You've heard us say that, Reef is growing and expanding, and it's been a really exciting time of growth at Reef. And when we bring everybody up, you will see what we're talking about because it is really amazing. So staff, interns, fellows, why don't you guys come up here? Come on up. So we have an amazing team of 15 staff. 14 of us are here. Jana Nichols, who many of you might know. Yeah, look at this. What else happened to you? Yeah, so this is our team. The only one who's not here, come on, move down this way because everybody's crowded against the wall. There we go. We have 15 staff. Jana Nichols, our citizen science manager, is not here because she just had a new grandbaby and we couldn't pull her away from that. We've got 4 amazing interns, education and conservation interns. We've got a couple of fellows on our staff who are interns who've stayed with us. And about half of this team, yeah, good job, Finley. Half of this team is based here at the Reef campus and half is distributed around the US. And we are so fortunate to have this great group of folks. And because now we're so big, I'm not going to take the time to go down and introduce each person and say their titles because I'll certainly mess it up. But hopefully you've had a chance to meet all of us. And if you haven't, please take some time to check in and talk. Yes, good job. We've had a lot of, everybody's got really interesting stories and backgrounds and fun journeys that's brought us all together here. So thank you all. Thanks for all. Yes, good job. You have anything to say, Finley? Yeah, you do. Okay, you hold on to that. All right, so thank you, everybody. You guys can sit now. Thank you. All right. Without further ado, let's hear about some fish stories. Rob has come to us. He's spent, I think, born bred Miamian, and then from the north part of the state for a while. He's described many different fish species, which is super fun. So he's going to tell us some stories. So, thank you.
Thank you very much. Everybody can hear me okay? Cool. I want to make just some brief prefatory remarks. So when Christy reached out to me, I was immediately struck with the thought that this will be an audience that will know more about the topic that I would present on than I will know myself. And so I have to do whatever I can to avoid that error. Don't come down here and tell them a bunch of things they already know. That said, I'm going to go ahead and make a statement right now that I know you know something about more than I do, which is you guys have something special here. This is super nice. I've really enjoyed getting to meet everybody today, and what you guys showed for, in tribute to Paul was just lovely, and your shared mission has clearly made you a family. That's special. Okay, so with that said, I am going to try to put a spin on reef fishes so that it does come more in my favor, and maybe I do know a little bit more about them with respect to them being dead and preserved in jars of alcohol. So the title of my talk is The Reef Fishes of Gainesville, Florida, How the Florida Museum Fish Collection Informs Our Understanding of Reef Fish Communities. I hope by the end of the talk, you all will have a greater understanding of how museum collections in general inform our understanding of natural history and of what's happening in the real world. And we'll try to focus that down as we go to speak specifically to reef fishes. Okay, but before we get to all that, how did I get to standing here before you today? And that has a lot to do with these two people. That's my father, Dick Robbins, on the left, my mother, Catherine Hale Robbins, in the middle. And they fostered in me an interest in fishes from a very young age, and they allowed me to keep aquaria. I think my first aquarium was actually a hang-on-the-back filter with guppies in it, right? Started very young. And I have an older sister and I have a younger brother, and they're both professionals in their own right, doing things that my parents are very proud of. But of course, we all know here that a fulfilling life is one that involves working with fishes, or at least working with the marine realm, and so I'm eternally grateful to them. So I went to the University of Miami, and then after I left the University of Miami, I moved to Gainesville, Florida. Never looked back on Miami, although to come down and visit. It's a great place. It's just not one that I can compete in, I don't think. So when I went to work for the Florida Museum, I started doing a lot of marine activities, a lot of going out on oceanographic vessels. I think this is on the RV Velos out of the Florida Institute of Oceanography. One of my introductions to marine fish collecting for museums and for research was on the RV Longhorn out of Texas. But my interest, my focal research interest, to the extent that as a staff scientist I do research, which is limited, is on non-indigenous fishes in Florida. And I'm happy to say I get to publish some works on that, particularly with my wife. Okay, so what are we going to do today? We're going to have two acts, right? I really want to talk to you all about the Florida Museum broadly and the UF Fish Collection. How many of you heard of the Florida Museum of Natural History? More than I thought. That's great. That's wonderful. We're going to talk about the museum, and then we're going to talk about the UF Fish Collection as well. That'll maybe be the first 15 minutes, and then the last 20 minutes, we'll talk about the Reef Fishes of Gainesville, Florida, what I've promised to the Reef Fishes of Gainesville, Florida. No, it's not these three fish. They just happen to be a little tripartite design that works well. Well, I thought worked well. Okay, so the Florida Museum is the largest natural history-based museum in the United States, probably in the world. We have four buildings on campus, totaling some enormous amount of square feet, but more importantly are the specimens within the museum, the specimens and artifacts. And there's 40 million specimens and artifacts, which are what make it the largest natural history museum based on a university in the United States, but also makes it bigger than many standalone museums, including the Academy of Natural Sciences in Philadelphia, the American Museum in New York City, which has 32 million specimens, the Los Angeles County Museum, we know where that's located, 35 million specimens. It's as large as the Field Museum in Chicago, Illinois, 40 million specimens. It is an incredible amount of information stored in the form of specimens and artifacts available to tell us about the natural world. We have an exhibit and special programs facility that you saw in that past picture there that was Powell Hall there on the left with the butterfly rainforest on the right. We're actually about to tear the front off of it on January 1st, and it's going to be closed for 15 months. So if you want to see the Florida Museum sometime in the next 2 years, go now. We have about 250,000 visitors annually. This is where we sort of get beat like a drum by those big metropolitan-based museums like in New York City. But for tiny Alachua County, that's actually quite a lot. And we have a number of specific programs, outreach programs at the exhibit hall, both for adults and for youth. And we're exceptionally proud, I think, of our outreach online and in person to K through 12 schools, representing in the last fiscal year 423 schools through our Scientists in Every Florida Schools program. We have strong internet and social media presence. You can search at Florida Museum to find us on YouTube, Facebook, Science Facebook, Instagram, all the social medias. Some of the more popular sites online are our Fishes in the Freshwaters of Florida Gallery, and we say it that way because we know in this peninsula marine fishes come in, so we can't call them exactly freshwater fishes, but you can encounter them. So we list and display a number of those marine fishes in that online gallery. There's also a book by the same name. Snakes. The online snake ID guide is very popular because, well, snakes are supreme beings and they're amazing, right? And if I had a little bit more fortitude and independence, I would have been a herpetologist, but instead I took the path of least resistance. So anyways, a lot of really good information on natural history. I mean, it just goes on and on and on. There's, the International Shark Attack File. I was just talking to one of your guests here about that. We have information on butterflies and pollinators, the fossil history of Florida. I mean, there's skeletal specimen ID guides, tons of information. But at the core, of course, are those 40 million specimens and artifacts. I'm just going to start saying specimens because I'm not going to talk about artifacts tonight. And to get at that information, the museum employs 31 faculty curators, which is also quite a lot. relative to those other museums that I mentioned. 22 collection managers on one of those. Most of the work, probably, I think these people would admit, is being done by students. And that's everyone from the 21 postdoctoral fellows in the last fiscal year, over 100 graduate students, and 150 undergraduate students. I'm not sniffling because I'm emotional that you guys have made me that way today. It's just very cold in here, so I do apologize for the sniffles, okay? So for 2023, 2024, faculty and staff chaired 93 graduate committees. So this is all student research. We used the specimens for a whole lot of coursework on campus. Faculty and staff taught 42 university courses. And I think most importantly, 118 undergraduate independent studies. This is the way we grab these young people early. We show them what a fulfilling life with the natural world could be like, and we take them away from that life of riches that they would get as an engineer or a lawyer or a doctor or whatever, and fill their bellies with joy and not actual nourishment. Okay, the collections are a global resource. Actually, one of the least enjoyable aspects of my job is shipping specimens out to the very deserving researchers that we collaborate all over the world. It's just very hard to do with all the restrictions on shipping specimens. But just as an example of this, this material as it's shipped all over the world and also within the United States is used in a number of publications above and beyond those produced by our in-house cadre of students and faculty researchers, which is about over 200 publications annually every year. But many more publications are produced based on the information that scientists get from these specimens by people external to the Florida Museum. Just for the fish collection in any given year, it's about 20 papers globally that cite the specimens by their catalog numbers in those papers. That's all stuff that museums have been doing for a long time. We're trying to be a modern museum in the sense that being modern means we can leverage these specimens for more information more quickly and more accessibly than ever before. One of the ways we do that is by imaging the specimens. All right, so when you image these specimens, whether legacy or recently collected, these advances in imaging have made vast quantities of visual information instantly accessible. And this promotes and expedites discovery. Researchers and laypersons alike can check IDs, extract data for research, or use the images for education. I look back on some of the old files in our file cabinets at the Florida Museum and see where people wrote letters to one another on typewriters and waited 3 weeks for a response just to get one little bit of information. It's incredible what we can do now with imaging. And we've been very lucky as the Florida Museum to be a leader in the space in getting the money for the Open Vertebrate Project, which has just recently concluded. There's a summary paper about what it all accomplished, but in effect, this was a 5-year federally funded program that successfully delivered free digital 3D vertebrate anatomy models and data to researchers, educators, students, and the public. Right? And the CT scanning is a non-destructive technology. It reveals a specimen inside and out. Its skeleton, its muscles, circulatory nervous systems, internal organs, parasites, eggs, stomach contents. Never mind all those times when people had to dissect a tiny little rotifer out of the stomach of a fish that was 10 centimeters long, or 10 centimeters, gosh, 10 millimeters long, right? With shaky hands. Now you CT scanning, scan it, blow it up on a screen this big, sorry, and rotate it in morphospace until you get the angle you want. It's incredible technology that, of course, we've had in the medical realm for a long time, but now we're leveraging for museum specimens to get that information out. And this is happening fast. People are getting a lot of information. Here's a nice diagram from that OVERT project showing how CT scanning and other types of imaging leverage these museum collections to answer questions about organisms and their environments. So we said nothing about specimens as vouchers for DNA extraction and amplification and some other ways they're used, but time is short and we need to get to talking about reef fishes eventually. But before we do that, I do want to talk to you about the fish collection specifically. We've been talking about the museum broadly. Now we're going to talk about the fish collection. It's massive. It's 2.5 million specimens of those 40 million specimens and artifacts. And they are preserved in, all these specimens are preserved in ethyl isopropanol, which requires a special facility that we imaginatively call the Special Collections Facility. It was just built, opened in August of 2022. So Ken and Anna and Ned have toured the fish collection, but that was in the old building in Dickinson Hall. So we're moving massive amounts of material from one side of campus to the other. This building holds now 60,000 gallons of alcohol. How many people have been in a Total Wine? I'll take that as a yes. You've been in a Total Wine. So the director of the museum spoke to the Total Wine manager in Gainesville, Florida, and we were able to determine that this is 3 1/4 Total Wines. Okay, that's how much alcohol is in there. Obviously, this building now has state-of-the-art systems that create the proper conditions for centuries-long specimen preservation, so we can hold these things in perpetuity, but also life safety systems that keep it from all going up in flames and killing the people that work there, and anybody in the zip code potentially. So. Okay, finally, we're at the UF Fish Collection. We've been moving it. This is the new facility. Where I am standing on the right, just to give you another idea of the scale and size of the fish collection, that is one of 58 aisles like that, internal aisles. There are 88 shelves to my right. There are 88 to the left. There's 6,213 total. Basil Williams, who quite frankly has been doing all of the work of this move, one of the best hires we've ever made, is standing on one of the end caps there. So there's 6 of those. There's a total of 64 of these 115-inch tall mobile compactor shelves, not only for the fish collection that we have, but for the fish collection that we will have and that we must keep building in order to answer questions about the natural world. We have represented in the UF Fish Collection 8,000 species collected from some 31,000 places across the globe over a period of 160 years. Okay, that's Act 1. Are we with me so far? We've talked about the Florida Museum. You now know what it is. It's specimens. It's specimens, and it's the people that get the information out of them and publish on that information and improve our understanding of the natural world. You all are keen-eyed, and so you know this is not a fish from the Western North Atlantic, but it makes an apt demonstration of an introduction to our next part, which is discovery and description of reef fishes. And this is a Nemateleotris, 2 species. There's Lavandula and Helfrichi, and it shows some of the subtle differences between species. Okay. When I started this talk, I had no idea I was going to be talking about my father as much as I'm going to be, but there was no way around it. All right, I don't need a way around it. I love my dad. He's a great guy. But if we're going to talk about the reef fishes of Florida, then we have to talk about the Miami School of Ichthyology, which Dick Robbins and his students built from 1956 to 1994. He graduated 31 PhD students and 14 master students in that time. These people assembled one of the most important collections of tropical Atlantic fishes from the surface to the ocean depths. This included many fishes from the Florida Keys, where work was conducted largely by the students, and some of the work that we're going to talk about in a little while will undoubtedly be familiar to this audience. In particular, the students Walter Stark, William Davis, Henry Federn, and Alan Emery did a lot of that work. Also located on Virginia Key, where the Miami School of Ichthyology was located across the other side of Rickenbacker Causeway during this era, was an important federal institution with prominent ichthyologists employed there. And together, these groups made what we talk about as the Miami School of Ichthyology, or what is addressed in the literature as such. Okay, a little bit of homework before we get going and talking about this part 1 of Act 2, which is, if we're going to talk about species descriptions, we need to know the rules. And the first thing to know is that there weren't really any rules from the time of Linnaeus' binomial system instituted in 1758 until about 1895, when people got together and said, we need a code, we need rules, and they established the International Code of Zoological Nomenclature. This is going to be the dullest slide we do today, but it's also going to have your one homework assignment, okay? And so basically what the ICZN put in as their goal, put forth as their prime directive, if you will, for you Star Trek fans out there, right? One organism, one name, right? And the scholarly work that describes that species and the year in which it was first described, you can think of that as the birth certificate for that organism. This is what the ICZN says on their website themselves. This is not me picking metaphors. This is what they go with. Now, your one bit of homework here that you're not going to forget is that the type specimen, right? The type specimen for any species description is the name-bearing specimen. Simple definition. People always get it wrong. That's it, the name-bearing specimen. Let's look at some examples. Yeah, for emphasis. All right, finally a picture of some fish. Yay, or a fish. And everybody here knows this fish, I'm sure. Yellow prow goby. Turns out it was described by Jim Belke, who was a close friend of my father's, and also, as it turns out, my godfather, because when you live in an academic family, colleagues and graduate students end up getting dragged to things like christenings and so forth. So Jim Belke was my godfather. So this fish was described by Dick Robbins and Jim in 1968, all right? Here's a nice photo from Carlos and Allison Estape. This is one of 90 species that Dick Robbins described. They were not all reef species, but 28 of them were gobies, and undoubtedly most of those were gobies. More importantly, as a collective, that Miami School of Ichthyology, all of those students, they described another 690 species. Now, there's some overlap there because people publish together and vice versa, but just to give you an idea of the scale of contribution to our understanding of what is out there and what I hope you all are seeing on a day-to-day basis. Now here is the type, right? This is it. This is the name-bearing specimen. Gobiosoma, Xanthropora, Belke, and Robbins. Something's changed. It's Elacatinus here, and there's parentheses there. Why? The parentheses tell you that there's been a revision and that the species has been placed in a new group. That's all, right? So if you ever seen that before, you didn't know what it was for, that's why, okay? Now, the type specimen, the name never changes. Now, Robbins made sure that all the type specimens for the species they were describing went to the Academy of Natural Sciences in Philadelphia, which is why you see A and SP there for the catalog number and why you don't see UF or UMML, which would have been the prefixes for the Miami collection, which was later transferred to the University of Florida to the collection that I manage with funding from the NSF. But knowing the importance of types, you need to have a type specimen as the name-bearing specimen if you're going to describe future species that are closely related or similar, or if you can do any kind of revisionary work, you really want to look at those types. So it's so important for the stability of nomenclature that the types are preserved, and Robbins knew that that collection potentially was not going to be kept, wasn't certain where it would go, that the types were all deposited at the Academy of Natural Sciences in Philadelphia. Okay, so the name of the type never changes. Here's another fish familiar to all of you, tessellated blenny. I don't dive, but maybe you can tell me, pretty common, right? Yeah, fish you see with some regularity. Not described until 1980 by two of Robbins' students, Bill Smith-Finez and Arturo Acero. Bill Smith-Finez has been one of the most productive students in terms of species descriptions from the Miami School of Ichthyology. He has described 127 species on his own, and I'm also happy to say that it is his 83rd birthday today, and he is watching. So, happy birthday, Bill. Okay, so Bill and Arturo described this species in 1980, and here's its type. And you see there's no change in the birth certificate, so to speak, right? No parentheses, no change in genus. It's not been revised. Bill would probably like to think it's never going to be revised because he nailed it, got it right. Now, you still have nailed it, you still have gotten it right if somebody rearranges the clades that we all think of as wanting them to be monophyletic, wanting them to be a shared common ancestor in all the descendants. It's still a valid species, but these things do get recategorized from time to time. But the type, the name never changes on the type. And here's just a brief picture of the description. Okay, we're going to be leaving this section soon, but we'll do one more before we go. And I chose this one, not because it's another Bill species, although you can't, swing the proverbial dead cat in a room of fish descriptions without hitting one of the Bill species. But, oh, this is a cat-friendly audience, sorry about that. I'm more a dog person myself, but so he described this species in 1997. This is a very common fish, is it not? So you see, it takes a long time from, and Christy and I were talking about this before, from discovery, from seeing something and knowing it's not described, to making it described. And so this was only described in 1997, and Bill remained very fond of his major advisor, Dick Robbins, and named it in his honor. Oh, going the wrong way. Okay, and here's the type. And now by now you guys know that name doesn't change, and there's not been any revision because the names are the same. Okay, and so all of this that we've been describing, that's the alpha literature. That's the basis for the general texts that are so useful to us all, particularly these identification guides, but they are the foundation for all this work. And when one of these general texts does not work for your identification purposes, the next step in the process is to go seek out that alpha literature and see if that doesn't work. When that doesn't work, either you're doing something wrong or maybe you have something new, right? And then you can go further and further down that road. Okay. Finally, something about fish diversity, because there's an interesting point to be made here as well, and I'm kind of interested to know if this is familiar to this group or not. But there's 36,953 species of fishes. That's very exact, right? Well, that's very exact because ichthyologists have their act together. And ichthyologists have their act together because another student of the Miami School of Ichthyology spent his entire professional career, along with writing books and publishing papers on scorpion fishes, building the catalog of fishes, which is a clearinghouse for all the published literature going back to before Linnaeus and since, anything you can lay hands on, and making sense of all those species descriptions, applying the code to them that you guys are now an expert in, and using things like the principle of priority to determine who described a thing first, what's the synonym, what's the name we used. And we have that all in a single database out of the California Academy of Sciences in San Francisco. And that tells us that we have 36,953 species. That's not the interesting thing. The interesting thing is fishes are the super diverse vertebrate clade, and yet we're still describing them today. What I want to know from this crowd is, do you know how many we are describing or at what rate? And so a quick quiz here is going to be, would you say of those 36,953, let's say in the last 10 years, have we described 1%, say 36, of all the fishes ever described in 300 years? Has it been, say, 330? Has it been 300? Has it been in the last 10 years? Has it been 3,600? Has it been 10%? Could it be as much as 10% in the last 10 years? I'm leading the question. Yes. Okay. Yeah, it's familiar to you guys. And in the last 20 years, 7,953 in the last 20 years. Okay. This process is far from done, is my point. Okay. We're still describing species. We're still learning what's out there. And I had to throw this in as well, just to sort of insult all the other vertebrates. And also, you see all these sources. It's a hot mess trying to get these numbers. So much so that this is the one slide that I carried over from a previous talk, and I wasn't about to try to find all this stuff again. There's competing lists for the reptiles. Okay, so now we're into Act 2 of the reef fishes. We're in Act 2, and we're in Part 2, surveys of reef fishes. All right, and right now you guys have realized that I am presenting nothing new that I have developed. I am simply the narrator of a whole bunch of things that other people have done. Let's talk about Stark 1968, a list of fishes of Alligator Reef, Florida, with comments on the nature of Florida reef fish fauna. What do you all think the chances are that Walter Stark was part of the Miami School of Ichthyology? Yeah, he was the first PhD student of Dick Robbins's, and he surveyed Alligator Reef, which you all know better than me, as an outer reef on the edge of the Gulf Stream offshore of Isla Morada. And he used every method available, including things that we would not conscience today, like dynamite, but also things like rotenone, which I know is frequently villainized today, but has a vanishingly small effect on reefs and on reef fishes, and is a very essential tool for getting at cryptic diversity. But he did that from 1958 to 1967. With a lot of help from his wife, I might add, and if you read that paper, you'll see how much that she contributed. And the only previous biological study before Stark did this to mention the area in publication was Breeder on the Pawnee expedition in 1925, and a paper was published 2 years that reported on 15 species. How many did Stark find? 516 species. These are not all reef species. He categorized 389 of them as reef species. And importantly, at least from my perspective, over 2,000 cataloged lots and many thousands of specimens, all placed in the UMML Miami School of Ichthyology Marine Fish Collection, which has been since transferred to the University of Florida. And that was, at the time, one of the most comprehensive surveys of a reef fish community anywhere in the world. And let's flash forward to 2017. And this is all familiar to you guys, I realize, right? So Carlos and Allison Estape were doing surveys of reef fishes, visual observations. They came along and they came to realize that this paper by Stark had been published. They contacted Stark and they said, hey, let's get to do that together and update the list, right? And so they did that and found through visual observations another 41 species, including 3 by reef, and another 66 from the museum records. So the fish collection, long after being transferred from Miami to the University of Florida, has continued to be growing and has continued to be updated and refined, and most importantly, digitized and available online so that you could rapidly get at this information. We're going to conclude the talk in about 10, 15 minutes in discussing how this is actually unfortunately not very common for natural history collections, and so this information is locked away and not accessible. But because it was online and accessible for Walter Stark and the Estapes, they were more readily able to report on these changes. And so this is what they found, right? They just went on our website, they went to our web portal and looked at alligator reef. They ran a query and said, okay, this is what's here, and then they downloaded this into spreadsheet format. This is the georeferenced stuff. But you know, the Florida Museum Fish Collection has a lot of reef fish specimens and not just from alligator reef. And so if you just looked at this area without the constraints of alligator reef, you'd see something more like this, which seems like a lot. It seems like a lot of fish murder, right? But it's not, okay? I mean, each one of those pins is like 50 miles wide, and each one of those pins may be one specimen of one species. It may be 50 species and 300 specimens, but it is an astonishingly small drop in the bucket. And they weren't all taken at the same time either. Remember, these are collected over decades and decades. But the Florida Museum does have a very strong collection of reef fishes from all over the Western Atlantic, including the Cayman Islands, the Virgin Islands, the Lesser Antilles, smaller numbers of reef fish collections from Puerto Rico, Jamaica, and it goes on and on and on. A lot of information to be had. And anybody can search this information using the web portal, and you can run whatever query you want using these criteria, these tables over here on the left. Here's one I just ran quickly to see what images we had of reef fishes. These were the dots for them. Historically, this information has been used largely by taxonomists, but that is changing, and that's what we're going to finish up by talking about. When Stark and the Estapes published their 2017 update, they produced a load of tables showing differences between the 1968 study and the 2017 study. And this was just one that I chose at somewhat random because I thought it was interesting, because this business of 59 formerly common to abundant species not seen or only rarely seen in 2017, to me, it's not even important that we know the answers to why they were more rarely, not seen as often the second time around, but that we can ask the question of the data, that we know based upon the specimens we have that there has been this change. And again, because at my heart, I'm a taxonomist, or at least somebody who works in the service of taxonomists, I like the fact that this table shows that those materials collected by Stark back in 1950, whatever it was, 57 to 68, were used as type specimens. H is for holotype, that's the name-bearing specimen, the single one. Paratypes are the other materials that a species describer uses and cites essentially as supporting cast, material that you could use to nominate a replacement holotype if it was lost. And you can see all of these species that were used in species descriptions since the 1968. Sorry, I should not touch my microphone. Oh, and I should mention, so then there has been an update of the update, which you all are probably more familiar with than I am, in 2020, with more visual observations and more changes in the database, mostly due to species splits, and now the total is 632 species. And the authors make a point of saying that it's only because other areas haven't been surveyed to that extent that alligator reef has this sort of unparalleled diversity. If you start looking at other areas and using museum reef-based collections as your basis and truth for them, you can see that the diversity is great in those areas as well. So one really interesting study is from the Bahamas, and that study, even though it doesn't mention the UMML collection or the UF collection in the title, has about the same proportion of those specimens from the UMML UF collection as the ANSP. And this other study I mentioned just in passing, because I thought, again, it was interesting that it brings home the point that visual observations are great. We need visual observations. What you all are doing as an organization is critical, but we also need to make other surveys of specimens to get at those cryptic or difficult to find fishes, or we won't know they're there, and we won't know whether they're there from one decade to the next, and if they're disappearing, why. We want to be able to ask the question, why. Okay, we're in the final stretch here, folks. Novel uses of reef fish specimens, the last part of Act 2, and I'm going to step off the ledge here into areas that I'm less familiar with, but are nonetheless, I think, compelling with regards to why we use museum collections to tell us about the natural world, tell us things about the natural world. Okay, here's a black sea bass. Why a black sea bass? We're going to find out in one slide. We're going to talk briefly about stable isotope analysis, right? And an isotope, going back to chemistry class, is each of two or more forms of the same element, but with a different mass, owing to different numbers of neutrons in their nuclei. Stable isotope analysis is basically determining the ratio of isotopes so that one can better understand the flow of energy to reconstruct past environmental and climatic conditions, but more relevantly to what we are talking about, the use of museum specimens that we're about to discuss, the flow of energy through a food web, right? And how has that been changing through time? So this is possible that stable isotopes serve our line of inquiry in this way because the ratio of the isotopes of a given element vary between different substances. That can be the food that animals eat, the water that they drink, and these isotopes are incorporated into the organism's tissues. That unlocks the ability to use museum collections to tell us about trophic shifts in the actual environment. So Dr. Madison Willert is a marine ecologist in the National Sea Grant Office who investigates trophic shifts in marine fishes through time. She does this by sampling museum specimens for stable isotopes to estimate a baseline trophic position. This particular study, published in 2023, was based on specimens at the Harvard Museum, one of the oldest and most valuable collections of specimens in the United States. And she and her co-authors looked at the black sea bass, which is a mesopredator, and the scup, which is a benthic omnivore, and looked at them over time using museum specimens, and then went down to the docks and got currently collected specimens and found a trophic shift, a trophic decline in these species, suggesting that the marine food webs in this area are being flattened due to heavy exploitation, with unknown consequences for these species as well as for ecosystem functioning. In Natalia's talk, she talked about animals as bioindicators. Another way of labeling these organisms would be as sentinel species. And these are excellent sentinel species to tell us about changes not happening only within their ranks, but within the overall food web as a whole. So before Dr. Madison Willert was Dr. Madison Willert, she was Maddie, and she came to the Florida Museum as a PhD student in 2019 and told me that she wanted to chop the fins off of hundreds of specimens, to which I did not have a particularly pleasant reaction at first, but she convinced me of the value of this line of inquiry, and she has a much larger study that is in preparation. She was kind enough to share this figure from that paper that is based upon specimens from the Florida Museum and closer to home based upon fishes entirely from the Floridan region. And they have surveyed many species now for these potential trophic changes and to be able to say what has happened. One of these species, and this is preliminary data, is the offshore lizardfish. And the preliminary results of the ratio of nitrogen 15 to nitrogen 14, or for us, the nitrogen stable isotopic signature, show a potential decline in the trophic position of the offshore lizardfish in Florida. So more use of museum specimens to tell us about what's happening in the world. Now, there's a little arrow showing a little diagram on the left. Forgot about that. Now, but if you go and make a clumsy, haphazard Google search like I did, Google Scholar, and ask things about like novel use, museum specimens, or reef fish and the like, you get a cascade of results, right? And all this to tell you guys something that I think you probably knew already, but maybe don't think about as much because you are working in the marine realm with living creatures, but that in order to tell us what's happening in the world, we need to go back in time and look at specimens in museums, and we can't do that if we don't have specimens in museums, and if those specimens aren't digitized in a way that you can readily get at the information. So we can use these museum specimens to tell us about increasing microplastic concentrations. I was pleased to see that some of these concepts were also recapitulated in Natalia's talk. Length-weight relationships. Well, that seems silly, right? Like, if you want to know the length-weight relationship of fish, you just go catch it and measure it and weigh it, right? You know, there's 37,000 odd species of fish. Most of them are really, really hard to catch, okay? And if you want to do a visual census of an area and then say the biomass of the living reef fishes in this area is X, what are you going to base that on? You're going to go out and collect one of every fish and then take that as a representative? No, but you could go to museums and with some coefficients of correction and a couple of little statistical analyses here, get a proxy for what it might actually be and do your analysis. Turns out marine parasites are declining in some areas, which runs counter to what a lot of people hypothesize, which is, well, as fish populations become more stressed and they become weaker due to all the anthropogenic impacts, then maybe they'd be more parasitized. Not necessarily so, at least by the results of one paper. You're going to study environmental DNA. Where'd you get that DNA from as your measure of the DNA you're getting in the environment? I hope it was from a museum specimen that you can go back and check again and actually see that it was correctly identified, or that you can correct if it was misidentified. I don't always identify things correctly. Changing climate, mercury burden, information on non-native fishes, an area near and dear to my heart in terms of my research interest, parasite species as non-indigenous introductions themselves. Okay, we're coming to the end right here in the next two slides. So this paper was just published in 2023, a global approach for natural history museum collections. And this is where I make my pitch, my final pitch to you all about the value of natural history collections. So these authors, and I agree, and they're citing a bunch of papers that say this already, and of course you know this now from this happy little presentation, global collections are the physical basis for our understanding of the natural world. They're an unparalleled source, directly relevant to issues as diverse as wildlife conservation, pandemic preparedness, food security, and the bioeconomy. These authors described a simple and fast method to assess the contents of any natural history museum. I didn't find it quite as simple as perhaps they put it forth to be, but they did underscore really important points about the need to further digitize natural history museum collection holdings. They tallied up over 1 billion specimens in museums, and they called for thoughtful coordination of future collecting efforts. And I think organizations like Reef would be really key in coordinating what things do need to be collected so that in 2100, we can answer questions that people will have then about what was changing and what was happening in the environment in 2024 and what those differences look like between the year that they're living in and the year and the specimens were collected. So finally, here's your take-home messages. Natural History Museum specimens are foundational to organismal and environmental knowledge. I beat that drum pretty hard. The type specimen, it's the name-bearing specimen. It's nothing more complicated than that. And I firmly believe, based upon the experience I've had in the last 28 years working in the Florida Museum, I've seen a lot of this change just in that very short time, that how museum specimens will be used in the future will be different. They're irreplaceable. They won't ever be something that will stand in for them. They're always going to be the ultimate source of the information. So I have to acknowledge a number of people. Mary Brown, my partner in a lot of this stuff having to do with fishes and just our shared interest in the natural world. My 24/7 support staff, Ruby and Willa. Another part of a fulfilling life is of course, dogs. And I will acknowledge cats. Cats are fun too. Carlos and Alison Estape for sharing photos. Kristen Grace and Zach Randall for doing the same from the Florida Museum. For recent help with published works, Carlos and also Madison Willett. And for the photos of those ANSP type specimens, I contacted the fellows at that museum that do similar to what I do, Kyle and Mark, and then of course the Florida Museum Departmental Travel Fund. Thanks. Questions? Yes, sir.
Yeah, question about papers describing the new species. Sometimes it's really hard to get those papers. Do you have any ideas as to how to get a hold of a paper describing a specific species?
Yes, you can do exactly what my wife does and ask somebody you know well that works at a university and has great access. But no, you make an excellent point. I run into the same thing. If I'm not on the VPN for the University of Florida, I just get blocked again and again and again. And this is an issue that is larger than anything I can solve because it comes down to money, right? And the whole publishing model is, in a way, I don't want to, it's not, yeah, I don't want to use that word at all, so I'm not going to say it. It's in need of reform, okay? And Scott's nodding knowingly, probably thinking about a bunch of different things, and I'll nominate one idea, one he might be nodding about, which is just that peer review is unpaid, right? And so we're relying on a lot of very busy people who are already stressed out to peer review these papers and do a rigorous and good job and not get paid to do it, which means they might not do as good a job as if their time was compensated for. But yeah, at the end of the day, the paper should be open access. Some journals do that now, and that has, I think, in large part to do with how much money the people publishing the paper pay in page charges up front. So yeah, the simplest answer is ask somebody with good access for now.
My question is similar. How does one access the OVERT project?
Oh, so that's great. You can go to morphosource.org right now, and now all of those shape files are available for free. You're going to need some knowledge and some software on how to take those files and then render them into a visual three-dimensional image that then you can use for whatever you want. That OVERT summary paper is great. Federally funded research, right? You paid for it. So we made a point, I say we, I wasn't involved directly with the program. Dave Blackburn was the lead. He's our department chair and herpetology curator. And he made it all available for free, but also made it by request because we wanted to know, how's it being used? So he'd get a request in the middle of the night from somebody that would say, hey, I would like to use this shapefile for Lophius gastrophysis, this type of goosefish. And my intended purpose is I'm gonna make an ashtray out of it. Approved, awesome, you know? And there's a lot of art being done with specimens as well. And just knowing how the information is used is great. Morphosource.org. Yeah, it's run out of Duke.
Thank you. I make reproductions of organisms.
Go wild.
So
yeah.
sounds like a great resource to me.
Yeah, and so that program was to CT scan an example of every vertebrate genus held in museums, and it was nationwide, 6 partner institutions with UF as the lead. A lot of fish and reptiles and birds flying through the mail the last 10 years. Yes.
Thank you. It was an excellent presentation.
Thank you.
So also a question about money. Where does the money come from this amazing facility and the time and the people and oh my gosh. So who do we have to thank for the investment that's been made in the work that you're doing right now?
I'm going to go with the old, there's a proximate and an ultimate answer. And I'm going to say the proximate answer, and this is not pandering because he's probably not watching. And also the both of us are going to retire in such a time that I can't be pandering. But Doug Jones, our director, during my time at the museum, has been visionary and effective. He's been incredible. Ultimately, most of the funding comes from external grants, right? And so all those faculty and staff and students, you know, students get their own funding these days. You know, students are applying for graduate school now. Sometimes the way they get into graduate school, besides all the wonderful accomplishments and all their smarts as demonstrated through their grades and their GRE scores and everything, is their ability also to write a compelling research proposal, sometimes with that future PI to get funded as they're coming in the door. The new building specifically, that got funded probably for the wrong reason, which was, for me, the right reason is, of course it gets funded. These are incredibly, incredibly valuable specimens. But if they were as valuable to society as we all think they are, because you're all on board with me now, right? And I definitely think they are, then that's where we would put our money, right? I just think that's what happened. But we got the money because somebody in the provost's office, presumably, or in some office, I don't know who, said, there's how much alcohol in that building? And that being the old building that had no fire suppression systems at all. In fact, we were under probation for a number of years with the state fire marshal. Also, again, kudos to Doug Jones for navigating that landscape. And just before COVID, the university went on a big low-interest loan spending spree, I guess, and a bunch of money was spent, and that building, which ran $13 million, was the cheapest of all the buildings they were on their spending spree for. Public safety was 37 million, engineering was 60 million, stuff like that. Yeah.
Using the drift poison method of collecting the specimens, how is it prevented from taking too many specimens from one particular type of fish so that you wouldn't negatively affect the population?
So what I would say as an answer to that is to reference a paper by Ross Robinson and Bill Smith-Inez concerning the topical use of rotenone in applied situations where you have expert persons such as yourselves that know about what species are likely to be there, what their biology is like, what the currents are like that day, and obviously try to administer the rotenone in a way that is likely to yield the most results in terms of discovering what species are present, and without doing more killing of fishes than you would otherwise intend to do. I think the idea these days, and I am not a diver, and I've never used rotenone, you saw my father happily applying it back then, is to use it as a tool to determine presence absence. And then also, sometimes to presence absence, discover new species and get some specimens of those new species. I'm not sure I have answered your question, and if I haven't, it's because I'm woefully deficient on the topic. But that paper, and there are a number of papers that I used as reference for this talk, which I'm happy to share and send to Christy, and you can disseminate. But I think Robertson and Smith-Inez 2008 is probably freely available. Or I'll make it so. You hear that, Bill? I'm giving away your paper. All right, that's it. Thank you very much. Appreciate it all.
Yeah, got to get out of my straitjacket. All right, thank you so much. That was amazing. That really interesting. And as someone who read a lot of those papers and thought about those when I was younger, that's really interesting. Well, thanks for joining us today. This was a really great afternoon with Natalia and Kramer, and capping it off with Rob, and of course, getting to spend a little bit of time thinking and remembering about Paul was really special. So tomorrow we're back at it again. We have a couple of great talks tomorrow afternoon. Our own Dr. Ali Candelmo is going to tell us all about the super amazing work that Reef is doing right now in conservation science, with a couple of fun technology projects. And we've got Stephen Frank, who's a very long time photographer here in the Florida Keys, who's going to tell us about the new normal in the Keys and the changes that we've all seen, and that's being exhibited throughout the Caribbean, and what that might mean for us and the natural world moving forward. And then tomorrow night we have our For the Love of the Sea, and the weather is supposed to get quite better, and we'll have a lovely afternoon celebration. If you're coming tomorrow night, you haven't bought a ticket yet, it's really helpful if you can do that in advance, so we're not have to try and scramble getting you registered at the door. But we hope you will join us. And yeah, I wish you well. Be safe out there, and we'll see you tomorrow.